70%. And it ended up at this, which is amazing. One amino acid completely changes the fold. So on the left-hand side, you've got 3 alpha helices, in the middle of which you've got 1 leucine, leucine 45. They make 1 mutation to tyrosine, and look, it's completely changed to an all beta. So in other words, they've made a metastable protein whose fold depends on a single amino acid. And right, this might sound like an exercise in spending taxpayer money and getting nowhere. But when they go back and look into nature, they found these metastable proteins. There's metastable proteins in the human genome that can take on 2 completely different folds, which kind of goes against everything you taught. You know, you don't always have to have an energetic minimum giving you a single structure. And this experiment proves that. Right, so quick word on resins, right? So any resin you buy for the purpose of doing purification is usually made, look at the quote marks, inert polymeric beads. So they sell you this idea that it should be inert with regards to binding your protein. The only thing that should bind your protein are the groups on the end of the beads. But this isn't actually that true, right? Your protein's quite capable of binding the material the beads are made of. They're porous to give you high surface area, so they've got high binding capacity. And the more homogeneous the size of the beads, the better your column will perform, but the more expensive the column will be to buy. And usually made of cross-linked agarose, cephalose. And your experiment will go something like this. It'll be really straightforward. You've got your matrix on the left, providing a charge, low strength buffer. So remember I said that will be somewhere between 50 to 100 millimolar, something like that. Enough to keep your protein happy, but not enough to compete out. And if you're monitoring the signal at A280, you've got a baseline you've equilibrated with the buffer here. You pass your dialyzed sample over, and your protein adopts a charge that's complementary to the resin, and it sticks. And then you'll see an upturn in the absorbance as the contaminants flow through and out the column. So maybe they have opposite charge to the resin, maybe they just mask the charge and they don't bind. Either way, all your contaminants are coming out here in the first band. Then, as soon as you've returned to baseline, and just buffers coming through then, you'll switch to a high salt solution, which will then knock your protein off the column. So your 2 choices, right, is either you do a step and you go from very little salt to high salt in one go, and that takes all the protein off the column, but that does it in a small volume, which can be quite advantageous. So a step will give you a dirtier sample in a smaller volume, or you run a gradient where the pumps mix the low salt buffer with the high salt buffer, and if you've got an ion meter on the machine, you can measure the conductivity increase over time, and then you hit a point at which your protein comes off, and hopefully that peak there would just be your protein of choice. And then next time you'll know exactly the right amount of salt to put in to take your protein off. So as I mentioned, you usually follow the A280 signal. If you've got no tryptophans, you shift that signal from 280 nanometers down to this range, 190 to 220, and you get absorbance from the polypeptide backbone instead. Also, though, the nucleic acid will influence that signal, so you don't want any DNA, RNA still present in solution. And you'll know when your protein's eluted, then you'll run a gel. And for example, what you tend to do then, so you've got peak 1, peak 2, peak 3. If you're running a gradient of salt up here, then you might want to end with a final step, and that washes the resin. So usually this will be about 2 molar salt, and anything still bound to the resin will come off there. And you've got this little signal here, and these are really tight binders, or maybe that's not even protein. Maybe that's small molecules contaminating DNA, something like that. So you collect your samples off the end of the column. You analyze it depending on what you're looking for. So SDS page is probably the easiest way to do it. You expect the band at a certain molecular weight. You might have to Western blot it if you've got a low amount of sample. You might have to run an activity assay if you're looking to, let's say you've got 2 populations. Maybe your active protein and your inactive protein come off at different times. And this happens quite often, right? Quite often in chromatography. The protein that's become inactive has different properties from your regular active protein. So it does help for you to perform an activity assay if you do have an enzyme. Then you retain your samples and you concentrate it, and usually then we now move to a different method of purifying it. We'll do size. We'll purify things based on their size rather than their charge. And so, you know, here's a few smaller examples like insulin, myoglobin versus something like myosin. And because they have different sizes, different hydration shells, different behavior, we could separate them quite easily based on their different properties. So the idea here is if we have a resin with the correct porosity, so our beads now are still made of an inert material, but we have these pores within, and the property we're looking for is to select a material that will allow our molecule of choice in and not allow anything bigger in. So, for example, the pore size here is selected so it lets the orange protein in, but is restrictive for the size of the blue protein. So if you had a packed bed of resin there, and you added your sample at the top, what we start off with here is what I would call our void peak, and the first peak you see come out the end of the column is stuff that's so big it can't interact at all. And as well as being big contaminants, this will often be mess. So misfolded protein, aggregate, all the bad stuff comes off first, doesn't interact with the beads at all, and you throw that out. And then over time, your larger species that interact weakly come out, and your smaller species, which get retained strongly, come right out at the end. And you can make a correlation between volume of elution and size. And what you'd normally do is you make the really poor assumption that all proteins are the same shape. So you standardize your column based on globular, compact proteins. So for example, you run cytochrome C down there, and that'll be about, I'll get it wrong now, be about 20 kilodaltons. Then you'll run something bigger, and you make a calibration chart. And so at the expected elution of your protein, you'll be like, I should be coming off at so many mils out. But that makes the assumption that everything is the same shape, and of course it isn't. So you get a bit of wobble based on that. The other thing you see, right, is the quality of your sample is reported by the shape of your curve. So if you get a perfect bell-shaped curve, you know your sample is lovely. But if you get a curve that has a shoulder to the left, that means your sample is aggregating over time and is becoming bigger over time. So what would you do then? If I had something that looks like this, I'm going to draw over it. If I got something that looks like this, I'd be well happy. And if I got something that looks like this, that is probably composed of two things. It's composed of one lot of that superimposed, oh, my pen's terrible, superimposed with one lot of that. And what I would do then is I would ask myself, is that bigger species something that just happens anyway? Like, does my protein have an equilibrium between a monomeric form and a dimeric form? Or is that bigger species because my protein is precipitating and aggregating and becoming unusable? And the way to solve that is to take the smallest right-hand peak and not collect the left-hand part of it. And then you take the right-hand peak, you concentrate it down, assuming it's the smaller entity, and you inject it back in. And if you get a lovely bell-shaped curve, it shows you that the two species are separate. But if you get the same curve you started with, it tells you your protein is in equilibrium. So this is the nicest thing you can do with your technique, is to take a peak and re-inject it and work out how your protein's behaving over time. So off it goes, and like, dependent on whether your lab is set up to do this and what you like to purify, you pick your resin based on the expected size of your protein and what you want to separate it from. So if you're interested in peptides, you'd be down this small end, and if you're interested in things that are larger assemblies, you'd be interested in this higher end. So you pick your, like I said, you pick your resin based on the pore size and what you're trying to actually enrich for and separate with. And look, it's massive, right? You go all the way from 10 to the 2 to 10 to the 8 in terms of size separation. So this is a big one for me, right? So that's theoretical. I gave you a theoretical curve. And I'm like, right, this protein's this size, this protein's that size. They will run differently. So that only runs true if everything is globular. But if everything's got a funny shape, then that's not going to be the case. Or, if your protein sticks to the beads rather than goes into them, it will be retained as if it was smaller. You see what I mean? So it's retention time. If it even remotely interacts with the beads, it'll look as though it's a smaller protein than it actually is. So what you would do then, so if you get this result that suggests your protein is smaller than it is, you would rerun it with higher salt, and you'd be trying to break the interaction between your protein and the resin. So quite often you'll run size exclusion like this in a silly amount of salt, in like 1 molar, 2 molar salt. And you'd be like, okay, does it run truer now? Does it run truer to its expected size? So raised ionic strength, or, right, that protein resin, size exclusion resin, let's say it's made of cephros, then that's a derivative of a carbohydrate that comes from seaweed. Now, what if your protein's a carbohydrate binding protein? There's enough of those about. So you'd be like, oh, maybe I should be using a different kind of material in my beads. So your properties, proteins are known weight, give you your calibration curve, and your volume is always kept low, right? You don't inject large volumes on, because then they're going to come off in too large a volume and you get no separation. The smaller the volume you can inject onto the resin, then the better your separation. And the thinner your elution peak is in terms of profile, which is usually, before this stage, why you concentrate your sample down to like 0.5 ml. But that also tends to aggregate it, so. Here we go. Here's another cool protein, right? Have any of you heard of the Volt? No? This is amazing, right? How can you not hear have an organelle present in eukaryotic cells that's 3 times bigger than the ribosome? And the reason why you haven't heard of it is because no one knows what it does. So here's an organelle present in eukaryotic cells. It has 39-fold symmetry
which is a beautiful thing in itself. And it makes this sort of doubly symmetric vase. As I've mentioned, it's 3 times bigger than the ribosome. If you look from the side, it looks like a vase. If you look top down, you can see that 39-fold radial symmetry. And then if we just take one of the monomers out, it looks like this. So this would be at the top over here. Then it curves round as a single snake in helix. Then it's got all these repeat domains in a row that fold to give you the body of the vase. And it's present in cells, and you can see it in electron micrographs. And we have no clue what it does. The only thing we know is if cells become resistant to cancer drugs, they make more of these. And sometimes they find the cancer drugs inside the vase shape. But it's amazing, right? There's an organelle present in biology, and we don't know what it does. And this is it. It's called the vault, which is a great name. Right, affinity resins. So the more modern way of doing things now, right, is to maybe use size and charge at the end, but begin with the idea that you can place a tag upon your protein that gives it specific affinity to a resin. And you can see here, this is a nickel histidine resin. And it's blue charged with blue from the color nickel. And so through genetic engineering, we can introduce our gene into any host and any vector of choice, and we can make sure that that vector of choice has an amino acid sequence that tags our protein. And so this is usually the way you do it. So our protein would be at the top there, and then at one of its termini would have a polyhistidine tag, and you see that in blue, and at least 2 of the histidines would make an interaction with nickel, which sits on the resin and is held by this spacer in red. And so our cell lysate would get poured over the nickel. Here we go. In this case, we've used 3 acetic acids as our ligand. So here's the bead here. You've got a spacer. That holds the nickel. Then our histidine comes in and completes the coordination shell. And all the other proteins flow through. And what you would be wary of here is there's a couple of common contaminants in different systems. So, for example, in E. coli, there's a couple of proteins we know also behave like this. They would purify too, but you get rid of those quite easily. And then our histidine interaction with the nickel is sterically competed. So we see a structure histidine here with imidazole, which is basically the histidine side chain without the linker to the amino acid part. And then imidazole will compete our protein off, and we'd see massive enrichment for our tagged protein. The only issue we have here, right, is that you should include 5 to 10 millimolar imidazole to reduce non-specific binding. So you spike your lysate with a small amount of imidazole, that gets rid of the contaminants. Your protein with a longer tag still binds, and then your actual elution is anywhere from 100 millimolar to 300 millimolar imidazole. The other thing you have to keep in mind with a his-tagged prep is that imidazole is actually denaturing. So what you might find is your protein binds the resin, you add this huge concentration of imidazole to knock it off, and then you're left with cottage cheese in the sample as the protein denatures. So what you would do there is you'd say, okay, I won't use imidazole to elute. You'd bind your protein on, it would bind the nickel, and then you'd either drop the pH to protonate the nitrogens on the imidazole, on the histidine. So you'd elute off at a lower pH, or you'd add something like 50 molar EDTA, and the EDTA would come in and it would take the nickel out of this interaction and your protein would fall off. So if you're doing his-tagged protein prep and your protein precipitates afterwards, then don't use imidazole. Shift to a pH or use EDTA.
And the reason why his tags are so good, you can start with a lysate like this, and in a single step, end up with an elution profile like that. Look, you got 1, 2, 3, 4 contaminants, and then you probably use a different chromatography method to clean that up. The other good thing about it, right, is that interaction between the protein and the resin is not dependent on the protein being folded. So you can do it under denaturing conditions. So if your protein gets made and then put into inclusion bodies, so if you've never come across inclusion bodies, it's like a waxy white material present in the cell when the cell can't fold your protein. So what you do is you purify that inclusion material away using centrifugation, and then you resolubilize it in urea or guanidine, and your protein's unfolded, but you can still bind to the nickel resin. So that's quite a nice way. And you get 100% pure, usually, but then you have to refold your protein. The nickel resin is cheap, and we recycle it usually about 20 to 30 times before throwing it out. And then the idea then would be you might introduce a protease site between your tag and your protein. So the cheap ones tend to be thrombin or sumo protease or something like that. And then you cleave the tag off. You cannot use EDTA as a protease inhibitor because I've mentioned that will take the protein off the resin. And often, right, the histidine tag will make your protein aggregate. So you can imagine if there's any spare nickel in solution, then 2 tags will come together and your protein will aggregate. So it quite often makes sense after to add a little bit of EDTA just to take any nickel out of solution or something like that. The other thing you can find with his tag proteins is sometimes the tag gets hidden. So if you imagine you've got a protein with a cleft and a tag on the end, sometimes the tag folds back into the cleft and it won't bind the resin. So if you put your lysate over the column and you see no binding, what you should do then is add a mild denaturant. So like half a molar of urea, half molar urea will make that tag fold out and then it'll bind the resin beautifully. Or, right, if you're purifying a membrane protein in a micelle of detergent, sometimes the tag goes into the micelle. And so what you would do there, instead of using 6 histidine tag, you make that unfavored. You make a 10 or 12 histidine tag, and it'll come back out of the micelle, and then you can purify your membrane protein. So I mentioned the denaturing. You dissolve it normally in urea or guanidine. Guanidine is better than urea, but is also refolding, I think, is harder in guanidine than it is in urea for some reason. And then you refold the protein, which is possibly the world's simplest sentence, but the thing hardest to do, right? If you've ever tried to refold a protein, it very rarely works. This is quite a nice way of doing it, right? So I don't want to push a particular manufacturer's product, so don't look at where it comes from at the top. But what you do in this case is you can buy beads selective for the his tag that are magnetic. So instead of doing everything on a resin, you add the beads into solution, you tumble it to allow binding, and then you use a magnet to draw the beads down. You just pour the solution off. And you've purified a protein there in about 10 seconds. So this is a quite nice way of doing it, this magnetic agarose. Or what you do here is we've got a set of roller bars, and in the top you've got nickel resin and your lysate. You can see it's a bit orange there. There's your cell lysate. So you roll it
just to increase binding. So you can imagine, right, if you've got a resin in a column and you pour your lysate on, its interaction is actually quite limited as gravity pulls it through. But something like this, it tumbles with the resin and mixes well, and you get really nice binding. And then these little tubes go sit in the centrifuge, and a really low speed spin will pull the liquid through this filter that's capped here. And so all you do, right, is you incubate your lysate with the resin in the top chamber, you put the cap on it, you roll it for 10-20 minutes, then you take that cap off, and there's a selective filter there, you spin it in the centrifuge for like 2 minutes, all the liquid comes through, and you're left with your protein on the resin, and then you add a little bit of imidazole, back on the roller, back in the centrifuge, and then you spin it and your protein comes through. So this is a really nice way of doing it. You don't even go anywhere near a column or a cold room or anything. Off you go. So not just his tags, right? You've got all these other systems now. The his tag is, as I mentioned, is usually 6 histidines, anywhere up to 12 histidines, or you can make a fusion protein tag that's bigger. So GST is 200, just over 200 amino acids. MBP is massive, right? It's adding a tag to your protein that's almost 400 amino acids big. So what happens then is you make a fusion of your protein with MBP, and this is written as a plus, but it's not. It says solubilize in effect. So you can tag anything to MBP and it'll get made. So in other words, you could tag the world's worst protein to MBP. It'll get made, it'll look as though it's soluble, and then when you cleave the linker between the MBP protein and your protein, the MBP will be fine, and your horrible protein will then come out of solutions. This is a very artificial way of thinking you've got success. So MBP as a tag is good, but this solubilizing effect here is a false positive as to how stable your protein is. You've got a strep tag, you've got a flag tag, and you've got these different eluting agents that then take the fusion protein off the resin. And you can imagine, right, maltose is a lot cheaper than buying a peptide. So depending on the amount of money at your disposal, it will affect which one of these you choose to be your fusion partner. So, you know, the pluses down here is that these two tags are massively specific, and the negatives are that the cost of the eluting agent is almost prohibitive to you using these. There's no reason why you can't have a protein with 2 tags, 2 different tags, one at the N terminus, one at the C terminus. So quite often in my lab, we'll have MBP at one end and his at the other. So you can have a look later at that. What's the problem with tagging? It is artificial. So like, for my purposes, if I'm doing structural biology, that doesn't really matter, as long as I can take the tag off and do structural work. But imagine if you're making recombinant insulin, then you don't want the tag on the end, because then you have to go to the regulatory approval of injecting insulin into someone's body with a tag on the end. You know, it cannot be tagged. Protein therapeutics used cannot really be tagged. So you need to remove it. And so like factor 10 protease will recognize these sequences, thrombin will recognize this sequence, and so on. TEV's pretty good. TEV's quite specific. And usually what you'll do in your lab is you'll have one expression strain that makes the fusion protein tag, and you'll have one other set of cells that maybe makes TEV, and you'll have a stock of TEV present in the freezer that you can then add without having to buy in. Because if you're doing this quite frequently, you don't want to keep purchasing these expensive proteases. Or, you know, if you really set up to do this, then why don't you have a tag on the protease? So then you can do a nice experiment where, let's say I've got a his tag coupled to my protein with a TEV sequence in the middle. If I put the his tag on the TEV, the protease itself, then I mix the two together. The protease cuts the tag off my protein, but is tagged itself. And then I put them back over the resin, and then my protein will come off pure. And I don't need to purify it away from the tag or the protease. So sometimes you use a tagged protease at the same time for that purpose. So I've mentioned the drawback there is the need to remove the protease afterwards, but if you tag the protease, then that's not even a consideration. Oh, this is an awesome protein, right? So I'm always a little bit surprised when you look in nature and there's nothing that's really blue, is there? You know, like peacocks and certain fish and that. And you think, well, why aren't things really that blue? And this is a blue protein. So here's crystals of it in solution. You can see how blue it is. And it's got probably one of the best names, Rana Smurfin. So it's named after the Smurfs, if you're familiar with how blue they are. And Rana is frog. And what it is, it's quite amazing. There's a frog that spits blue spit. And then it makes a nest out of the spit. So this frog's nest is blue. And the reason being that the main ingredient of its spit is this ranasmurfin protein. And if you look at it, so we're zooming in on the active center of this, it's got a tyrosine and a histidine that have become covalently linked into this sort of clathrate structure here. And so it's used normal amino acids, but in such proximities that they now covalently bind each other in an activated sense. And then sat in the middle is a metal ion, and all the distances around that spectroscopically is what gives you the blue absorption. So you can see all the connectivity there between the side chains. And what's awesome, right, is they're like, why does it do this? Why does this frog make this blue spit? And the reason is, this frog's eggs are massively sensitive. You know, there's no shell on a frog's eggs, obviously. They're massively sensitive to UV radiation. So by making a blue nest, it protects the eggs from UV DNA damage. Very clever. Proteins are brilliant. Right, so in terms of tagging, right, I just let you know that you've got to get rid of the tag and you've got to have a protease system. And sometimes for pharmaceutical products, therapeutic proteins, you don't want that. But what if you had a system that was self-cleaving and left no remnant? So in other words, your fusion protein had a protease within it, and that protease could be selectively activated to excise your protein of choice. And this is amazing. This is this natural intine system. So you know about like self-cleaving pieces of mRNA or whatever, introns, exons, etc. But it happens in the protein world, and it's called an intine. And it's very natural, and you can see it in a few proteins. For example, it's present in type 3 secretion proteins. And this middle part is flanked by a cysteine and an asparagine. And under different redox conditions, so in this case we're using DTT. So in other words, we've got our fusion protein, we bound it to the resin, we add DTT in, and what DTT, this is DTT here, it comes in and it breaks this linkage, and in doing so, you've now got activation at this end, and this cyclizes, and it kicks the protein out. So any tag you want, you know, if you want, you can put your protein over here, or you can put your protein here, but a 3-partner N-hexene, intene, C-hexene, the whole fusion protein here has self-cleaved, look, leaving nothing on the end. So this is a really nice system. It's hard, it's very difficult to get large amounts of protein using this system. But what you do end up with is something with absolutely no remnant of the tag on the end of it. And this will only be optimized further, right? Because you find these situations in nature. You know, maybe we'll end up finding a thermophilic version, which would be lovely. Now, this is a really clever idea for tagging. So, like, most of the tags I've shown you are very, look, this is what it is, this is the property of the tag, this is how it would help you do something, right? But imagine, again, I'm going to draw over it, sorry. Imagine you were working on a membrane protein receptor, right? So you can excuse my appalling drawing. Let's switch pens here. Imagine you're working on part of a transmembrane. Here's transmembrane number 1. Here's a loop that we're interested in. Here's transmembrane helix number 2. So let's say this was a receptor present in humans, and we know all the goodness happens at this end, but that peptide part is tethered in the cell at the membrane for purposes of signaling, right? And if we couldn't make the membrane protein, because it's often difficult to make membrane proteins, but we wanted to study the binding properties of this receptor, what we'd have to do is we'd have to clone this region out as a separate entity, which sounds easy, but maybe its folding is dependent on being held in 2 places, right? So like, as a biochemist, the first thing I would do is be like, all right, let's find ourselves a protein that has 2 helices and a loop, and we'll switch it out, right? We'll use 2 soluble helices in place of the 2 transmembrane helices, and then we'll just make a soluble version of our receptor. And this has been done enough times to render that approach doable. But this is an amazing way of doing it. I love this way of doing it, so I'll go over this slowly. And this hasn't really taken off, but it's ingenious. It's very clever. So that receptor loop there is now going to be our test protein in this case. And we know that we want to tether the 2 ends, but we don't know the best way of doing it. So what we do is our test protein in red, which might be stable, it might not be stable. We haven't quite got there yet. We clone it in between 2 linker regions, shown there in grey. So we've got our red receptor, 2 linker regions. Now, N-terminal flanking, we place half a beta-lactamase. C-terminal flanking in light blue, we place the other half of beta-lactamase. Now, if our test protein is good, and we've designed it correctly, and it's stable, but we don't know that, then the stability of it will allow the 2 ends of beta-lactamase to come together and be a beta-lactamase that's functional. And then you can select for this by expressing it in a cell with beta-lactams present. So in other words, if the beta-lactamase is functional, it'll turn over the beta-lactam and the cell will survive. So you've got selection pressure. So this system now has been engineered in this way. You might want to use error-prone PCR to give you the variance of the test protein. You might want to use different construct ends. But either way, it gives you a selection pressure that allows you to select directly for a stable protein, which is absolute. It's just genius. It's very, very clever. So we can do that now. So elements of different proteins, for example, our receptor, we could clone them in, and we could find a stable folded variant using this method. Lemons, right? So this machine is beautiful in the top left there. So this is the Prometheus machine. And what this does is it measures unfolding of protein in real time. So the old method for doing this, right? If you want to keep a protein stable, I draw over my lemons here. What you would do is you've got a dye, and this dye, if you've ever come across it, is fluorescent. It's called Cipro orange. And what you would do is you find that Cipro orange is only fluorescent in a hydrophobic environment. So, in other words, your protein folds its hydrophobic core, and your fluorescent signal from this is low. So you place the dye in with your protein, and you measure the fluorescence. But if your protein unfolded, then this dye now has a nice hydrophobic environment because it sees the inside of your protein. It gives you a fluorescent signal. So what you can do is you can put this in a qPCR machine, and its temperature will raise in increments, and you can measure fluorescence. So you can measure protein unfolding in real time, which is related to the fluorescence signal. And so people have been doing this now for quite some years. And what you would do is you would run these experiments in different buffers. You'd run them in different salts. You'd run them in different additives. And you'd find something that shifts the... So the unfolding curve will look like this, right? So you'd have fluorescence there. You'd have heat. And you get a curve like that. And then this is the half. This would be the temperature at which half your protein starts to unfold. And you'd look for a condition in which this curve was shifted this way. In other words, it's more stable. And you can do this now, and this is a very successful way of finding ways to stabilize your protein by measuring the difference in unfolding curves. What this machine does is it does exactly the same thing, but in tiny capillaries, so you don't need much sample. And it does it not by monitoring the fluorescence of a dye, but measuring the fluorescence of those tryptophans that are present inside your protein. So you've got to have a tryptophan. And we've been using this, and it's a really nice way of doing it. And the reason why I have lemons up there is because almost every protein you put in is very responsive to citrate. Why is citrate so good for proteins? It's because it's a natural metabolite present in the cell. So proteins probably see okay-ish concentration of citrate. Anyway, citrate's highly charged and stabilizing, and it's a good buffer. Right? Citrate has 3 pKa's because it's got three carboxylate groups. So citrate is a very good buffering agent for proteins. So your classical strategy, old strategy, would be to lyse your cells from your expression, ammonium sulfate precipitate out all the gunk, dialyze it overnight so you can reduce the concentration of that ammonium sulfate, then select on a different principle, in this case ion exchange. Your sample will come off in a pretty wide-ish peak, which is a larger volume. So you'd concentrate that down using a spin filter, and that smaller volume would allow you to inject based on a third principle, gel filtration on size. And you'd be very unlucky not to end up with a homogeneous, purified sample at the end. So a more modern way of doing things, go very recombinantly, find an expression system that maximizes the starting amount of your protein. You do an intermediate purification until your protein was the dominant band, as judged by SDS page. You check using an assay that that dominant band of your target was still active, was still good, was still folded, was still biologically relevant, then you'd run one of those stabilization assays where you'd say, okay, in which buffers and salts is my protein happiest? Then you could rerun this under those conditions. You could concentrate the sample, you could add your protease to digest the tag off, and your final stage is still classical. You do a gel filtration chromatography. That void peak on the left would get rid of all the parts of all the variants of your protein that are unfolded. Because you've optimized the buffer and selected based on size, you then, on the right-hand side of your gel filtration, you'd hopefully have something with a symmetrical curve which shows you that your protein is now in a singular stable state, and then you could use it for all your downstream applications.
Right. So I will put these slides up.